Okay, so today's topic is third week of development. Previously, we have discussed the first and the second weeks of development. Today, I'll discuss the third week of development. So, third week starts with the process of gastrulation. What is gastrulation? Gastrulation is the process of formation of the three germ layers, which are the endoderm, mesoderm, and ectoderm. It is the process by which the three germ layers, which are the precursors of all the embryonic tissues and axial orientation, are established. So, in the second week of development, the bilaminar embryonic disc was formed. Bilaminar, the two laminae were the epiblast and the hypoblast. Now, those are being converted into a trilaminar embryonic disc, and this is the process of gastrulation. So, the epiblast and hypoblast will convert into ectoderm, endoderm, and mesoderm in this third week of development. So this is the bilaminar embryonic disc, the epiblast in blue color and the hypoblast in yellow color. These two layers in the second week of development will convert into three layers, the ectoderm, mesoderm, and endoderm in the third week of development. So what is the process of gastrulation? What happens is on the embryonic disc, there is a structure that is called the primitive streak. It is on the surface of the epiblast. On the cephalic end of the primitive streak is a structure that is called the primitive node. This elevated structure is the primitive node and the central depression is the primitive pit. So this line is the primitive streak. Cephalic end is signified by the primitive node and in the center is the primitive pit. Process of gastrulation is such that the cells of the epiblast they start invaginating in this primitive streak. This blue layer is the epiblast layer. The cells start detaching from this layer and starts invaginating into the primitive streak. This is the process of invagination. Now, a few cells come to lie, they displace the hypoblast, this yellow layer, and they are now called the endoderm. A few cells penetrate between the epiblast and the hypoblast, and this is called the mesoderm layer. And the remaining cells of the epiblast, they will form the future ectoderm. In this picture, we can see this slipper-shaped structure is the embryonic disc. This structure is the primitive streak. This is the primitive node surrounding the primitive pit. Cells from this epiblast layer starts invaginating and thus they form the three layers, the ectoderm, endoderm, and the mesoderm. As I've told in the previous slide, the first process is the process of invagination of the epiblast cells into the primitive streak. Those cells displacing the hypoblast are called the endoderm. The ones that lie between the epiblast and the hypoblast forms the mesoderm, and the remaining epiblast forms the ectoderm. Now there are three areas where ectoderm, endoderm, and mesoderm, the three layers are not present entirely. Only the ectoderm and the endoderm is present, mesoderm is absent. These three areas are this cephalic portion, the oropharyngeal membrane, this cloacal membrane, and in this region there is a process called the notochordal process. So these three areas are where the ectoderm directly comes in contact with the endoderm. So the fate of the primitive streak is that it degenerates eventually. In the embryo, it degenerates by the end of the fourth week of development. Now, there is a structure called the sacrococcygeal teratoma. As I've told you that the primitive streak degenerates eventually. But if it persists in the sacrococcygeal region, it leads to a formation of a teratoma that is called the sacrococcygeal teratoma. Teratoma is a tumor that has the tissues of all three germ layers, the ectoderm, endoderm, and mesoderm. The incidence of sacrococcygeal teratoma is 1 in 37,000 births. Now we'll discuss what is fate map. Fate map is this embryonic disc, this slipper-shaped embryonic disc is the epiblast. Now fate map is that the regions of the epiblast are mapped. They migrate and ingress through the primitive streak, and further they are mapped and their ultimate fates are determined. So this is called the fate map. So the mapping of the epiblast invagination into the primitive streak is called the fate map. So this round structure is the cloacal membrane. This is the area of the primitive node. So the cells that migrate from the cranial region of the node will eventually form the precaudal plate and the notochord. This notochord and the precaudal plate. From the cells that migrate from the lateral edges of the node and from the cranial end of this streak, they will form the paraxial mesoderm. This area is of paraxial mesoderm, bluish area. Now the cells migrating from the mid streak area, this purple area, will form the intermediate mesoderm. The cells from the caudal region of the streak, this light green structure, they will form the lateral plate mesoderm. And the cells that will migrate from the caudalmost part of the streak will form the extraembryonic mesoderm, this yellowish structure. So fate map is such that the epiblast cells that will migrate from the node and the subsequent structures of the primitive streak will be mapped, and they will form the various structures which are the notochord, the paraxial mesoderm, the intermediate mesoderm, the lateral plate mesoderm, and the extraembryonic mesoderm. This is fate map. Now, what is notochordal process? Notochordal process is when the pre-notochordal cells, the epiblast cells which are in the notochordal area, are called the pre-notochordal cells. They invaginate in the primitive node and move forward cranially, forming a process called the notochordal process. This area is the notochordal process. This reddish structure is the notochordal process here. In this picture, we can see that this blackish structure in the C picture, you can see that this circular structure is the notochord. The notochord will be formed by the notochordal plate, which will be formed by the prenotochordal cells and the prenotochordal process. Now, what is prenotochordal process? What happens is that the prenotochordal cells, they start migrating cranially, then they intercalate into the endoderm and form the notochordal plate. After that, they detach from the endoderm and form a structure that is the future notochord. The extent of notochord is from the oropharyngeal membrane in the cephalic region till the primitive node. And the function of notochord is that it leads to the formation of the neural plate here, and it is a signaling center for the axial skeleton. Fate of notochord is that it will degenerate except for the structure that will form the nucleus pulposus. Now, what is nucleus pulposus? The vertebrae have a small structure in between which is called the intervertebral disc. The marginal portion of the intervertebral disc is called the annulus fibrosus, and the soft central structure is called the nucleus pulposus. So, this nucleus pulposus is a remnant of the notochord. As I've told you earlier, that the notochordal tissue is almost entirely degenerated. If it retains, it leads to formation of benign or malignant tumors which are called the chordomas. Now, what happens further in the development of trophoblast in third week? Till second week, we saw the formation of the primary villus. What was primary villus? Primary villus was a central trophoblast in the center, which was surrounded by a layer of syncytial trophoblast. This happens till the second week of development. In third week, mesodermal tissue starts penetrating inside the cytotrophoblast, so it forms a secondary villus. So, mesoderm surrounded by cytotrophoblast surrounded by syncytial trophoblast is called the secondary villus. After that, tertiary villus is formed, which is when the villus capillaries start penetrating inside the mesoderm. So the villus capillaries in the mesoderm surrounded by cytotrophoblast surrounded by syncytial trophoblast is the tertiary villus. Tertiary villus is the definitive placental villus, and it is responsible for it is in its core are small blood vessels. Now, what are stem or anchoring villi? These are the villi that extend from the chorionic plate to the decidual plate. This is a stem villus which extends from the chorionic plate to the decidual plate. So this is all about today. Thank you.